Thank you.
And in 2019, she joined Shahar's lab. All of these three have a connection with Shahar. So that was very interesting. Where she was studying disordered proteins, but then in 2022 joined QSB and became a member of the Nobile lab, focusing on fungal pathogenesis using a planarian model to study this. She is a 6 times graduate fellowship awardee through the NSF-CCBM. He serves on UC Merced's IRB and graduate group executive committee, so doing a lot of service to help us with our program, which is great. Co-founded the Women in Science Special Interest Group here, and then also has maintained that K through 12 outreach during her graduate time. And outside of science, if that is not enough, that's what she's been doing. She enjoys rescuing dogs, exploring new interests, a little retail therapy, and also singing karaoke. So invite her.
Yes. Retail therapy is great. Thank you so much, Dr. Shepherdson, for the introduction, and thank you all for being here and supporting the trainees. We appreciate you. Yes, we're all bound by Shahar in some way, this group of presenters. So that's really nice. I did invite him. He's a little too busy in New York, but that's okay. This one's for you, Shahar, if it's good. Okay, so yes, I'm a fourth-year PhD candidate in the Nobile lab, and I'm going to shift us into thinking about fungal pathogens and preclinical animal model and how we can think about fungal infection in the context of in vivo infection. And so my title is New Insights into Fungal Host Pathogen Interactions from the Planarian Model. And so I'll start us on the fungal side of things. Oh, here we go. Fungal. Okay. And so we're surrounded by fungi, and many of us who are interested in fungi know this, right? Fungi are in the land, sea, and air. There are millions of different species among us, and many of them serve important ecological and world purposes, right? And some of them are decomposers. They help recycle nutrients in our world, which is important. Some are plant symbionts, so help recruit biomass and nutrients to plants, which helps their biome. And some have antimicrobial properties. Ever heard of penicillin? It's won the Nobel Prize. It's a fungus, right? So fungi can be very good. And the good news, out of all these millions and millions of species, very few harm humans. Bad news. Of these few, they can be quite severe to humans. Okay. And so the WHO issued a priority fungal pathogen list back in 2022. So this may have shifted over time, but I'll share with you what they described. And this was to help focus global research efforts into what are our most emergent threats in the fungal world. And so the criteria they based this list off of is a number of things. And some of these criteria are case fatality, annual incidence, global distribution, antifungal resistance, and any available treatments. And that's how they ranked the pathogens. And so the ranking follows as this. There's low, medium, high, and critical. Sorry, that's covered. We'll uncover it a little bit. Okay, and critical, right? Fungal pathogens on their list. Now, I'll briefly mention a few medium priority pathogens, fungal pathogens that are considered not terrible risk, but we should keep an eye on them, like coccidioides and cryptococcus, right? These are some examples from the WHO list. High priority, major health burden, Candida glabrata, tropicalis, and cropsilosis. Keep an eye on these. These can get you sick. And what about our critical priority group, the greatest worry we should have in terms of fungal pathogens? Candida auris, of course, Candida albicans, and Aspergillus fumigatus. A few of these are key fungal pathogens we need to be thinking about in terms of global health. And this is just a snapshot. It's not a comprehensive list, but of course you could check out the WHO full booklet. It's very nice. And so of these critical pathogens, I study Candida albicans, right? Couple key reasons why. Most of us have Candida albicans as a part of our normal microbiome, and that's totally fine. Most of us will be okay. It's thought to be a commensal, meaning it won't harm you, it won't necessarily help you. It's just kind of there. However, in the immunocompromised, Candida albicans can be a huge problem, and we know this to be true, especially in the hospital setting or in the setting of HIV/AIDS Candida albicans can take over and have a very high mortality rate in these individuals. Important to note, even healthy people can experience Candida albicans overgrowth, and most of us will at least once in our lifetimes, especially women, unfortunately. Okay, and it has a huge economic burden, right? Not just in terms of people getting sick, but even trying to treat this. It's in the billions of annual direct medical costs per year in the United States alone. This is just direct costs. Indirect costs, it will exceed this greatly, especially if we consider the global burden. And of course, Candida albicans can develop antimicrobial resistance. And some scientists argue this will be our next global pandemic, antimicrobial resistance. So we really do need to study Candida in the lab so we can figure this out. And so I'm curious as a researcher, I want to know, how does Candida albicans infection occur? And can we stop this infection? And so how do we study this in the lab? There are previous models that I'll mention briefly, and then I'll introduce our new model. So some previous models, which are useful and good models, but we need to think a little bit differently sometimes too. We have in silico modeling, of course, and this was talked about briefly. We have in vitro cell culture. We also heard a little bit about this today for other applications, of course. We have invertebrate model systems, right? So animals without spines. And then we have vertebrate animals, such as murine models. These have all been used to study Candida albicans infection previously. And they're all good models, but like any model, they all have pros and cons we should consider. And so in silico modeling, it's great, it's powerful, it's fast. You can do it anywhere in the world. You just need a computer, right? Some potential cons when thinking about in vivo infection is, can it necessarily recapitulate every biological complexity? Probably not, right? And of course, we should validate hits in vivo in terms of translational or clinical applications. Not to say it's a bad method. And same with in vitro cell culture, right? Probably more humane, cheaper than using whole animals. And we can gain insights like molecular and cellular level things, high throughput, could screen hundreds and hundreds of different conditions at a time, of course, but cons, of course, to like any method. Immortalization complicates things. We don't have a full whole body response from cells out of their native context. And invertebrates are nice too, right? We could study conserved antimicrobial response. We could use more animals compared to vertebrates, maybe cheaper. You could keep invertebrates in your house. It wouldn't even matter. You don't need a special facility. These are some potential pros. I'm not saying I've necessarily done this, but I won't deny it either. Just kidding. And some cons, right? Of course. Of course, invertebrates will also have some cons too, and especially with some where we can't necessarily dissect specific organ responses because it's all mashed up together and too tiny to study. Some have very poor tools just because maybe the field is still emerging. Maybe one day they will have better tools. Some just quickly succumb to fungal infection. There's nothing but a live dead readout. You can't study progression very easily or clearance. And what about our vertebrate models? They're very cool. They're very good. You can study them. They're thought to be a lot more like humans relative to insects and things like this in terms of a physiological response. And there's great imaging and histological techniques. And of course, there's some cons, right? Why don't we all use mice? It's difficult, it's expensive. There are ethical and regulatory considerations. Some genetic variability, perhaps. And in the Candida field, at least, they've been typically used for late stage fungal infection. So we're curious about early stage too. So it may not be the best model for us. And so enter the planarian. And of course, caveat, I'll mention this. All methods have good and bad things about them. None are perfect. So I'm not, trying to bash anyone's favorite model. I love them all. Love them all. Okay, so now the planarian, an alternative model system to in vivo infection. So if you haven't seen a planarian, it looks like this. They're found in freshwater habitats. Some people think they're very cute. They've got cute eyes. Some unique pros. They have a very good, robust innate system. So I'll talk more about that. They're clonal, asexual. You don't have to worry about buying more necessarily. You don't have to worry about genetic or differences. A sequenced genome, so we can learn a lot about the genome in planarians, and we can model infection pretty well. So hopefully I'll convince you of that today. And so planarians in general, there are thousands of different species globally from the land and air, and they've been described by humans actually. Oh, sorry, not air. Atlantic Sea. What did I say? Did not mean air. Who knows? Who knows? Maybe we just haven't discovered it yet. Won't rule it out fully. And so humans have been describing planarians for hundreds of years. This is some ancient Japanese text from the 1600s describing this land planarian, saying if you touch it, don't touch it, it's poisonous, but also it fissions when you touch it. So hopefully that person who wrote it is okay and survived. Now, this was mentioned a little bit earlier. I think Thomas Hunt Morgan came up. So planarians have been studying the lab for hundreds of years. So this means we have a lot of good tools to study planarians. And most, probably most famously, they've been studied by this Nobel laureate who studied them for their regenerative properties, which they're most well known for. And this might be a present day sort of regeneration study where you can amputate any part of the planarian and it will regenerate in an order of days. So you can see this blastema formation, which is this lighter sort of ends to the planarians, represents where tissue is being regenerated from where it was missing, from either purposeful amputation or any kind of injury. And like I said, planarians, we have great visualization techniques. They do have some anatomical complexity. We can study the multi-system response to infection. And like I said, they've got a great conserved innate immune system. I'll explain more about that later. But overall, planarians can be useful to help us understand the conserved antimicrobial response. And then on the pathogen side, they can help us understand what is the pathogen doing during early infection to infection progression and potentially clearance or lethality. So we can study both. So I'm telling you a very brief summary of the past 4 years of my work. Okay, so this took about 4 years to get working. Okay, so let me tell you briefly. How do we infect planarians? First, we start with some fungal culturing, of course, right? We have to plate it, grow it up overnight. That takes about 4 days. And then we have a fungal exposure period. This goes on for 3 days, where we introduce the cells directly to the planarians. So we know the quantity of cells we put in, and we know that it's a uniform distribution because we simply add it to the water they live in. And we expose for 3 days. And then we end the infection. And luckily in the system, it's easy to end the infection. It's not an intravenous model, which could be a little more complicated. And then at the same time, we're doing host outcome analysis. What does the host look like as it's undergoing this infection? And so there's a variety of different ways we can measure different endpoints that I'll explain to you. But in brief, we can study infection progression. We know what we're putting in, easy to stop, and we can have many different endpoints, unlike other invertebrate models where they typically just die. Planarians don't just die. Okay, so what's the first way we kind of analyze planarians as they're undergoing infection? So first, I'll show you what a healthy planarian looks like. This is a control, uninfected. It's swimming very, very fast. And then we have a planarian that's presenting with mild symptoms following infection. And these are they look sad, right? They're more compacted. Typically, planarians don't like light, so this one doesn't care if it's in the spotlight getting burned. It won't react, which is abnormal. And then we have animals that could present with a severe phenotype. And that could be a number of different ways. It could have wounds or lesions. It could experience head loss or eye loss or total fragmentation of the body. And then, of course, our most severe endpoint, which is death. So planarians can succumb to infection, but most of them don't. But I'll explain more about that. So this is to say planarians can display a range of clinical manifestations, and they don't just die. So that's very useful for us to study how this infection is happening. And so in case you didn't remember all of that, I made a very handy dandy flow chart to help researchers diagnose their planarians. And this will be in an upcoming publication that I will briefly chat about. And so that's great. We see planarians react to Candida, but what does this mean exactly? What does this mean for survival, for example? So we've been able to see that planarians, their survival will decrease in a dose-dependent fashion. So first I'll show you the survival percentage for uninfected planarians. It's always 100%, and they also don't show any symptoms, but I'll talk to you about symptoms in a sec. Okay, and each dot represents an aggregate of 5 animals that underwent either mock infection or a real infection. Okay, and then here I've added 70 million cells per mil into the planarians. And you see median survival is not really touched necessarily. That's no different. And then I add 80 million cells per mil. I add more. So median survival drops to about 50% in this case. And then I add even more Candida. I add 85 million. and the survival drops even further down to about 30%. Okay, and there are some statistical relationships between control and some of the subgroups here. And the point is to say here that it's not only the presence of Candida that affects survival, but it seems to be the amount as well. And it's in this dose-dependent fashion. And now we don't have to just look at morbidity or mortality, as I said earlier. We can look at both or even one or the other alone. So here I'm showing you between these two conditions, considering a lower and a higher dose, what fraction of animals are, for example, asymptomatic after undergoing this infection. And in this case, it's about the same in the two different groups, right? About 25% or so. Now, what if we consider symptomatic disease? Which percentage of animals are symptomatic after infection? And here we see some divergence between these two experimental conditions. Okay, now what if we look at how many animals are dead, right? And then we see further divergence between these two experimental conditions. At this lower dose, about 50% of the animals are sick. And at the higher dose, about 50% are dead. So this is very useful for virulence related assays, which is what I'm interested in. Which doses give us which kind of host outcomes. So this is informative in this way. And so planarians, as I mentioned, have a great conserved immune system. They don't just die. So what do? How does clearance look like? And it looks on the order of days. So here's a control animal. I'll show you immunofluorescence images. And here we're tagging Candida with a fluorescent label, okay? And so in the control or uninfected animal, there's no signal from Candida, right? So that's good, no cross-contamination. That's good. Okay, and what about an animal that's been infected and now we're looking 1 day post-infection? This is when we tend to see peak colonization as indicated by these clusters of Candida on the surface of the planarians. And so what if we keep looking some days later? And at 4 days post-infection, we see noticeable clearance actually, not full clearance yet, but noticeable clearance over time. And at 7 days post-infection, we see the majority of the planarians have cleared the infection. And this is consistent with other kind of research done in the infectious planarian disease world, where with bacterial or mycobacterial challenges on the order of 6 to 9 days, planarians can eliminate fully this infection. So they're very good at clearing different types of infections. Now, fungal infections is what we've established here. And so we're also interested in survival as well and kind of looking at it differently. For example, with the Kaplan-Meier curves, we can think about when do the planarians die and to what extent. So this here I use the 80 million cells per mil, that higher dose, where we saw about 50% death, right? And we have that exposure period. And we want to know when do they die and to what extent. And so the Kaplan-Meier tells us that around 4 days post-infection, we see the last drop off or the last event of death here. So that's useful to tell us. And it's pretty stable phenotype after this. Survivors tend to make a full recovery after this time point. So this is useful information to us. And so this is a research in progress. So now we've got the system working, right? We've got wild type working. So now we want to understand what's going on during this infection at a genetic level, and can we disrupt infection? This is what we're curious to use the model for, right? And so I have RNA sequencing data from the host and pathogen, the gold standard to understand which genes are involved in the specific process. This process is infection. So we have this information. We have candidates for knockout. If we knock out these genes, can infection be changed? And so I've been able to screen the top 10 differentially expressed transcription factors of the pathogen to understand how are these transcription factors affecting virulence at these different levels. So now I'll determine which are hits and perform kind of follow-up studies to understand why or why not these were hits. And then perhaps we can add antifungal to the equation and think, can antifungal agents rescue planarians that would have otherwise died without antifungal treatment? And then, of course, we can compare it to these different animal model systems I mentioned earlier. Are any of these hits conserved, and why or why not? These would be interesting questions I'm looking into in the last year of my work, okay? And so in summary, planarians, we say, are an effective alternative host for fungal infection. We can study disease progression, clearance, and lethality. Useful. We've developed the conditions for which to study these different endpoints, right? And of course, I'll add caveats, many caveats, of course, right? We know animal model systems are complex. So if you take this planarian to some other institution, I can't guarantee how the planarian will behave. So we have a lot of caveats in my new protocol that we've established where we, with full transparency, full data, we explain how to do this infection in many details. Okay, so after 4 years of development, now we can kind of apply this model in more relevant ways and learn more about fungal infection in vivo. Yeah, I'd love to hear what you all think, especially the animal folks. If you have any ideas on how to improve the model, maybe things you're curious to see, and we can work together and do that. So I'd like to acknowledge the labs I work with, the Nobile lab, the Oviedo lab, and the Sucinic lab, who I've worked previously with, but they still very much support me and are very excellent, excellent friends and colleagues to this day. And then absolutely thank you all for coming and listening. Happy to take questions if there's time. Yes.
Do your planaria that recover show like immunological memory in the sense that have you ever done a reinfection?
That's such a good question. So the question was, are planarians that survive infection, do they have immunological memory and maybe fare better if they're reinfected? I don't think we've tried this yet, but that'd be really interesting. I do think planarians have some primitive memory system, so perhaps they could survive better after reinfection. That'd be a really cool experiment. Yes.
I was just curious, I'd love to hear more of the backstory in the caveat of the colony replicability of your results. I mean, that sounds really, I'm super glad and impressed that you are bringing that up and thinking about it, so.
Thank you so much. I really appreciate that. So Dr. Ardell mentioned, thinking about variation, like colony to colony variation, and perhaps if we use it at different institutions, could the results be different? And I would suggest that yes, I absolutely think that animal variation is important to consider, right? And so part of this protocol, part of the motivation is how can other researchers kind of recapitulate what we've done in a reliable way? Because previously, I don't think this was possible, right? Especially in this model system. So think about preclinical animal models and how we could be more reproducible, right? And so in our protocol, we define some troubleshooting and initial setup steps. So how you could perform your own dose-dependent curve for a specific colony in your care, right? Because we all care for our animals slightly differently, even within the same lab. So kind of considering these things, I think could help other researchers be a little bit more consistent. So thank you for bringing that up. Yes.
I have a follow-up to that.
Yes.
Are you suggesting that the variation is colony management and care, or do you think it's more genetic drift from once you bring your animals from wherever you did your postdoc and you propagate them in your own lab?
That's such a good question. So the question is, what could it be accounting for this variability? Maybe handling care or perhaps genetic drift over time. I think that's a great question. I wish I could tell you. I do think handling and care is a big part of the equation. Unfortunately, I haven't necessarily looked at, what's the particular microbiome of this colony versus this colony, for example, or what's the gene expression of immunological markers here versus there. I think we could answer or sequence them as something happening. In theory, the planarians should not be changing their genomes because they're clones, they're genetic clones, but who knows, right? Should see sometimes maybe we should consider sequencing them maybe every 1 year, 5 years, 10 years, make sure things are looking okay. So I think both are important. So thank you. Thank you, please. Yes.
So is Candida part of the normal microbiota then for planaria? And why do you think then just these high dosing are causing problems that otherwise aren't?
Such a good question. So Candida, I don't think is not endogenous to the planarian. It lives in these aquatic settings, right? So it can encounter different ascomyotas, different types of fungi, but particularly Candida albicans, probably not. And so probably none of the animal preclinical models have Candida as part of their normal microbiome. So we are kind of introducing this weird novel bug. But they do exhibit a good response, at least some of these animal model systems, to the Candida infection, and they can clear it to some extent with many caveats I would add. But that's a good question that I think about too.
Good. I think that's all the time.
Okay, thank you.